Hello, everyone. My name is Luz Miryam Gomez. I am part of the team of the Coleccion Entomológica Tecnológico de Antioquia. First of all, I want to thank NATS and the symposium organizers for allowing showing deeper collections as one of the first steps to establishing collaborative work. The biological collection has served as a keystone for research and biodiversity education process. The Coleccion Entomológica Tecnológico de Antioquia has not been the exception. We want to use this meeting to share our experience applying research and student training during the last years. The Coleccion Entomológica Tecnológico de Antioquia belongs a public university institution located in Colombia, Northwest Medellín. It was established to store and deposit specimens collected in the field work from bioforensic research group, forensic entomology projects. In 2013, the collection obtained the National Registry of Biological Collections. As a research group, we also have permission to study biological diversity and access to genetic research. In this way, specimen deposits are legal from Colombian environmental authorities. We are aware of the taxonomy renewal and the importance of training young researchers to contribute to mitigating the taxonomic impediment. Also, we aim to contribute to entomological diversity studying in the Neotropical region and to the development of the application of forensic entomology as well. We started with a drawer containing 10 cornered boxes. Now we have an adequate facility that allow us to hold approximately 124 boxes under controlled environmental conditions. We house specimens spined or preserved in alcohol and their DNA samples. We manage all the information, data, and biological records in a specified software. Our specimens are not just a physical element. From there, information is derived that contributes to the knowledge of biodiversity of these taxonomic groups. For example, we have DNA sequences, we have morphometric information, we are made evolutionary analysis, description of new species, covering all the extensions of an extended specimen. We take care of a specimen from all biogeographic regions of Colombia, from anthropocene environments to pristine Amazonian and Chocó rainforest. Most of the samples were collected using Bansomer ride-on traps, baited with viscera of chicken, cold liver, fish head in the composition. The specimens from the Andean region were collected monthly over 3 years. We have approximately 89,000 specimens of flies of forensic importance, of which 37% have been able to determine the species lived. We work with California, Fanidae, Mucidae, and Sarcophagidae. In California is where the identification process is most advanced. The specialist of this group, Dr. Eduardo Amat, is part of our work team. In the beginning, Eliana Buenaventura, a specialist in Sarcophagidae, described new species of the genus Oxysarcodexia, and still more species remained undescribed, as well as the Inusidae. All the processed material is being sent to the biodiversity system information in Colombia and to the Deja Vu platform. In FANIDE, Jessica Durango, now the collection manager curator, began her process as a student. And for more than 10 years, she has focused on FANIDE identification and describing 7 new species. She expects to find more new taxa. Also, in addition to the taxonomic identification process, DNA sequences has been obtained, a species distribution model has been generated, and ecological niche modeling. Also, has been explored the use of geometric morphometry as a species identification tool. Students and young research professionals are linked to the entomological collection to support activities such as mounting samples, digitalization, doing manuscripts, publications as part of their formative process. A guide has been published which illustrates activities such as specimen collection and DNA extraction to facilitate the performance of the members of the team. The collection also supports undergraduate and master academic programs. The information derived from the research process has generated modules and subjects within academic curricula. And it also a space for a student to do or to develop their degree projects. Videos and didactic elements as, like trivia games have been developed to learn the process of an entomological sample, including fieldwork, collection, storage, preservation, identification, and forensic analysis. This entire process is guided by the researchers associated to the collection to follow the appropriate standards. Researchers from Brazil mainly have visited us, and we have exchanged training process and entomological material. As an goal, we pretend to make the information available through remote access and develop 3D models as an educative and taxonomic tool, understanding that web initiatives and technology are necessary as long as they aim to improve the existing taxonomic company, not to replace it. We are focused now in a new and interesting subject that promises outstanding scientific contribution by integrating forensic entomology and road ecology. Finally, with thanks to the Tecnológico de Antioquia for the institutional support, to the Ministry of Science and Technology of Colombia for project fundings and young research requirement and training, to the JVIF to support through for the support through Bid Caribbean Call. And finally, this is an invitation for you to visit us and establish work networks. Thank you and you're welcome.